As for Deb, the server. Yeah. We'll tell her that was her. That was not in place of tipping her course, but all right. Good evening, everyone. We're so excited you all could join us. That applause was for you, Deb. All right, we are so glad you all could come. My name is Chenry Fife. I'm the South Central Alaska Engagement Manager at TU. So Trout Unlimited is celebrating our 20th anniversary of conserving and restoring freshwater fish habitat in Alaska this year. So we're very excited about that milestone. We've been at the forefront of fighting pebble mine in Bristol Bay and ending old growth clear cut logging in the Tongass National Forest. We have been working to protect or advocate for salmon and wildlife on the proposed West Susitna Access and Ambler Road routes. Bristol Bay is facing lawsuits, so we are defending current safeguards in court, as well as working on more durable safeguards at the state level, specifically with House Bill 233 that was introduced last spring. So we'll keep you updated on that. We are also monitoring other threats to fish habitat around the state and in South Central. So last summer, public lands were under attack at the federal level, and we organized almost 100 businesses to protect those public lands, which actually worked. They were not disposed of this year. We also are monitoring things like the West Susitna Access Road. ADA has recently applied for their wetlands permit through the Army Corps, and we will keep you updated on comment periods around the West Sioux Road and anything else that comes up. We also have a lot of restoration projects active around the state right now. We are currently restoring 2 miles of Chinook salmon habitat on Resurrection Creek in Hope. We are also working with the BLM to protect and restore fish habitat on their lands in the interior. And we're working to reconnect fragmented salmon streams in Southeast Alaska and in Seward, as well as trying to bring back salmon to the Eklutna River. We also hired our full-time education coordinator last year. So we have some awesome youth events that we have been able to host and get involved in this year, including Adopt a Trout, which we partnered with Fish and Game, to teach middle schoolers about fish biology, telemetry, and trout in their own watersheds. And then also we brought Mayfly Project to Alaska this year, which pairs a fishing mentor with youth in foster care, which some of you have been volunteers for, so we appreciate that. So we often tell our supporters and TU members about these types of volunteer opportunities, and so we'll continue to do that. We also are getting involved in some invasive species mitigation work this upcoming year. So keep your eyes out next spring for some Elodius speakers and education, as well as a Matsu Pike Derby early next summer. So we want to thank you for your support and everything you do for wild fish in Alaska as well. You can follow us on social media or become a TU member or renew your TU membership. To hear about streamside planting days, future family fishing days, river cleanups, and that pike derby next summer. So feel free, as after the presentation, to walk over to that table. We've got some information you can take, as well as some stickers. The QR code helps you renew or pay for your membership, TU membership. It's just an annual small fee to support some of our work. And the first 8 people who sign up on the email list or for a TU membership get a free TU mug. So come see me afterwards as well. And I'd like to pass it to Pete Baldi, our South Central chapter president. Thank you.
All right, hey, good evening, everyone. It's great to see such an awesome crowd here tonight. I really had to put this together at the last second. It really just get it together. I didn't know how it would turn out, but it's excellent. Chenery just outlined a whole bunch of stuff that the Alaska TU is doing. South Central chapter is also hosting different events. We have next week, we have 2 barflies events. If you haven't been to that, we just show up at a local business, and usually it's a brewery, or in this case, peanut farm sometimes, and double shovel. We will just sit around and everybody ties flies, and it's just sort of a casual event. There's no structure to it particularly. It's kind of just a little get together. So next week, Wednesday evening, there'll be one. It'll be a women's barflies. It's going to be held over at the Double Shovel. It starts at 6:30 p.m. Anna is going to be the host for that. And so if you're interested, show up. But again, like I said, it's a women's only event. Then Thursday evening next week, that's the 18th, we'll be hosting a barflies event right here in this room again. It'll be at 6 o'clock. And like I say, you just bring your own equipment. We do have equipment and materials too for new tires. If you know anyone that wants to learn, send them over. There's always people willing to help and teach. And then we have one thing coming up next January. I'm working with Mark Drury. We're setting up Bristol Bay do-it-yourself fishing trip. And it'll be an informational presentation on getting your own fishing trips out in Bristol Bay. That's not completely set, but when you do know, you'll get it in the emails. They'll go out and you can find out then. Friday that just passed, we had an event in Wendler School where we went in and we helped teach kids how to fly cast, and it was quite interesting to see the range of skill that was shown. We spent a lot of time untangling reels. There was a lot of light. But anyway, but that was really interesting to see some of the kids who suddenly, when you go through and they teach them, just show them a little bit, and all of a sudden they get it and their faces light up, and you can see that someone has just absolutely fallen in love with the sport, and that you're going to see them out in the river in maybe 10 years. You know, they were all, it was really something to see. So anyway, we got a lot of events going on like that. Let's see, we have some other things going on. I don't know what else we got happening. I remember. So along with this, we have in the past, part of what is bringing our speaker here tonight is that a few years ago, we put in some money along with the Kenai chapter to extend the weir that's down on the Anchor River and capture the real late season steelhead run. So we can get a full year's data on the steelhead population on that river. And so after that, there was kind of a little push, and now we've gotten some word about this new program that's going on. And so one reason we got the speaker tonight is because we're really interested in raising some funds in order to help support the research that's ongoing. And so anyway, that's kind of a little introduction for our speaker tonight. Andy Seitz, he's a professor up at the UAF College of
Fisheries and Ocean Science.
Fisheries and Ocean Science. We go backwards. Fisheries and Ocean Sciences. They've been conducting research for about 10 years now, and they've been working in Southeast and also some up in the Bering Sea past Nome area on some Arden or Arden Char there. I'm sure, but anyway, he's going to hear from him tonight, and he's going to be introducing some new research a lot closer to home in this talk. So anyway, I want to introduce it. This is Andy Seitz.
I'm loud enough here. I'd like to thank the organizers for inviting me. And they actually didn't invite me, they invited Michael, but Michael's actually given a conference or a presentation. There's a biennial steelhead conference that brings people from Alaska and the lower 48 and British Columbia. And so he's giving a presentation on our work at the Steelhead Conference. And so I'm here My name's Andy Seitz. I'm a professor of fisheries at University of Alaska Fairbanks, and I live in Fairbanks, and so it's nice to come down here where it's a little bit warmer than it is in Fairbanks. Michael lives in Juneau, and he and I have been working together since 2012. And I'm going to tell you today about salmon and satellites. And you can see these are 3 salmonid fish, and they have a tag on them that communicates with satellites, hence the name of the talk. I was invited to give a talk, well, it was advertised that I was going to talk about steelhead. And then Pete said, well, maybe people would be interested in Chinook salmon too. So I decided to add in some dolly varden char in here as well. And so you're getting 3 for the price of 1 tonight. It's best deal of the year. So Anna's my trusty slide forwarder. So you want to go, there we go. So a roadmap, I have to, like when I teach a bunch of 19 and 20-year-old kids in college, and I ask them if they've ever seen a real roadmap, and most of them have not. So I like to say, here's my roadmap. I'm going to talk about the background of our work, the motivation for the satellite tagging, which fish we study, what we're trying to learn, how we do it, a very high level overview of what we found, and then the discussion is sort of how we interpret those results and what they're useful for. And then I'm going to spend a little bit of time on future directions, like Pete said. We're always looking for partnering with other organizations and studying other fish species in different places. So I'll talk a little bit about that. So a little bit of background. This probably isn't a surprise to most people here, but salmonids are found throughout North America, and they're really important. And so salmonids include the salmon, the chars, the whitefishes, and they're important throughout the state for a variety of reasons, including subsistence and commercial fisheries, like those 2 pictures up at the top. And then some people are probably familiar with that picture in the lower left there. That's the biggest king salmon ever captured on sport gear. That was captured in the Kenai River, weighed 97 pounds. And then they're important beyond just humans. They're important in ecosystems as well. And so they're abundant consumers. There's a lot of salmon in the ocean, and they eat a lot of food, and they can alter food webs. And then they're important prey too. And so one of the most iconic sort of predator-prey relationships that people think about are killer whales and king salmon. But other marine mammals eat salmon, sharks eat marine salmon, and a bunch of fish eat marine salmon as well. So next slide. And most research so far on anadromous salmonids, and anadromous, I'll tell you a little bit, but the punchline of the term anadromous is they're born in freshwater, swim to the ocean to feed and get big, and then swim back to freshwater to spawn. And that's what anadromous means. And most of the research on anadromous salmonids has been focused on the freshwater juveniles and the returning adults, because they're much more accessible in rivers than they are out in the open ocean and the high seas. They're kind of a needle in a haystack in the high seas. And then when they're in fresh water, they're in much more sort of a funnel, and they're easier to get access to. And so relatively little is known about their ocean ecology. There's the current knowledge of their ocean ecology. So when they're swimming out the ocean far from shore is limited to fisheries dependent data. And so we rely on fisheries to capture information about those fish. And so this can come from boats conducting research projects out the ocean. And that hasn't happened for quite a long time in US waters. And there's only one high sea survey left in the Pacific Ocean on salmon, and that's in the Central Bering Sea, conducted by the Japanese government. We do learn about salmon from bycatch in commercial fisheries, and so they're frequently captured in trawl fisheries. And so we learn about where those fish are captured and when. And then coded wire tagging programs, if they've got a coded wire in their speeder from the hatchery when they release, then we can get 2 points. We get a release location and a recapture location. And then we can also take these fish captured as bycatch fisheries and run genetics on them and get an idea of what their river of origin is. And next. But what's not known is stock-specific migrations and ecology of different stocks is not well understood. And so what we get are these looked a little clearer earlier. What we get are these sort of conceptual figures about what salmon do in the ocean. And so you get this sort of broad, high-level overviews. And so these are conceptual diagrams of how steelhead behave when they're in the ocean. And so you get these broad, like summer areas and winter areas with some arrows connecting them, but there's not a whole lot of detail. So next slide. And as a result of this coarse knowledge, there's a lot of uncertainties about ocean ecology of salmon, including their fine skin movement, population-specific migrations, diving behavior, habitat occupation, and how they die in the ocean. So, and this information can be important for, well, it's interesting, one, but it's it's also interesting in an applied context and can be used for thinking about a lot of management and conservation issues. And so it's important to understand this information for understanding overlap with human activities, and I'll give you some examples of that coming up. Population dynamics, so we can use this information to think about why salmon are abundant or not abundant in the ocean. For bioenergetics, and that is sort of estimating the fish's energy budget, how much it needs to eat to successfully grow. And in a changing ocean, we can use bioenergetics to make predictions about how salmon will respond in a changing ocean. And then susceptibility is bycatch. And so probably arguably the most high profile conservation issue in Alaska fisheries is bycatch in trawl fisheries. And we can use information about their ocean ecology to try and reduce bycatch as well. So, and so some examples of overlap with human activities. So each of these, so here's a fun fact, I'm colorblind, so I can't tell you colors. I just know that there's some squares on here, and those are oil and gas lease areas. So those are Bureau of Ocean Energy Management oil and gas lease areas. And so we can think about like how might a Dolly Varden char that swims into the Chukchi Sea up there in the northwest there, how might they swim through those gas lease areas? Do they swim through there? And if they do, when do they do that? And how might that coincide with exploration activities like seismic testing? Next slide. And then another example of human activities is this is a very blurry Kodiak. Here's the Kenai Peninsula. And this polygon here is called the TMAA. And you're going to see that later on. And that's the U.S. Navy's temporary maritime activities area, and the Navy calls activities blowing things up. And so what they do is every 2 summers they send a bunch of subs and surface warfare ships up to this area, and they don't have subsurface explosions, so the explosions are not underwater, but they have bombs up that go off in the air, and they are curious about how they interact with fish and marine mammals in this area. And so that's another example of human activities. So, that was sort of the background on it. Our motivation is to study so far 3 species of salmonids, and these include Dolly Varden char, which is in the top there, steelhead, which is in the middle, and Chinook salmon or king salmon down in the lower picture. And these fish are broadly distributed along the west coast of North America. And each of these species has several life histories. And life histories are, examples are how long they spend in freshwater, when they swim to the ocean for the first time, how long they spend in the ocean, and then when they swim back into freshwater as adults, and whether they survive spawning or whether they die after spawning. And so life history is sort of an animal's life story. And each of these species has variable life histories. But all 3 of them are anadromous. So we're going back to anadromous. And anadromous simply means they're born in freshwater. They swim out to the ocean to feed, and they return to freshwater to spawn. And we are focusing. So Dolly Varden and steelhead have a strictly freshwater form. Dolly Varden and rainbow trout. We are going to focus on the anadromous form. Yeah. So a little bit of background about each species, and I think Michael made this slide here to make it look confusing because Dolly Varden are very confusing. The bottom line of Dolly Varden life history is that they do whatever they want, and it makes them difficult to study. Here's their range up in the top panel there, Alaska in the east and Asia over in the west there. We're focusing on the anadromous form. But typically, and there's exceptions to all of this, but typically they spend 2 to 4 years rearing in fresh water, and then they swim to the ocean to feed, and they only occupy the ocean during the summertime, which is different than what you'll see for steelhead and Chinook salmon. And that's because the ocean where they feed gets really, really cold and they freeze like a popsicle in the ocean. And so they only spend a few weeks to a couple months out in the ocean, and then they return to fresh water in the fall to overwinter. And when they mature, they spawn in natal streams. And so they show homing just like Pacific salmon do, but only during their spawning months. And so in non-spawning years, so they only spawn every other year. In non-spawning years, they can go to any other freshwater system. And then Dolly Varden, anadromous dollies are idiroparous, meaning they are not pre-programmed to die after they spawn. So some of them do die after spawning just because they're worn out or they got no gas in the tank. But they are not pre-programmed like Pacific salmon is to die after spawning. Next up is Chinook salmon, also known as king salmon. Nice range map from, oh geez, I can't remember who said. And they are anadromous. They spend up to 2 years in fresh water, but like over 90% of the little king salmon in Alaska spend a year in fresh water before swimming out to the ocean. They swim out to the ocean and they can spend anywhere between 1 and 5 years at sea. Typically, the real big ones have spent 5 years out in the ocean. The medium-sized ones have spent about 3 years, and the little ones spend about a year. And so that's variable. The return migration timing is variable. So some of them, there's in Alaska, they're generally summer run fish, but elsewhere in their range, there's spring, summer, fall, and winter run Chinook salmon. And they are semoparous, which is the opposite of idiroparous, meaning they are pre-programmed to die after spawning. So 100% of them keel over and become bear food or seagull food or river fertilizer after they spawn. Yeah, and then the last fish we'll be talking about is steelhead. The steelhead is the anadromous form of rainbow trout. It's the exact same species. And they typically spend steelhead typically spend 2 to 3 years in fresh water as little youngsters. And then they swim out to the ocean and they typically spend 1 to 3 years out in the ocean. And they do spend winters out in the ocean, unlike Dolly Varden do. Their return timing varies. Most steelhead in Alaska return either in the fall or the spring. There are a couple summer run steelhead runs in Southeast Alaska. And then down in the lower 48, there are winter and many more summer runs. But they're all spring spawners. And they are also idiroparous, meaning they are not pre-programmed to die. But there are relatively very few second-time spawners. So we think about both Dolly Varden and steelhead being repeat spawners. There's not many of them. So they're not pre-programmed to die after spawning, but there's also not that many second-time spawners. So, and so the objectives of our research is we wanted to learn more about their ocean ecology, and specifically their movement, behavior, occupied habitat, and survivorship. And what we're primarily after, mainly because of funding constraints, or I shouldn't say constraints, because of funding interest, is spatial overlap in human activities. And I'll give you examples of those. So, and to study the ocean ecology of Dolly Varden, steelhead, and Chinook salmon, we use pop-up satellite archival tags. And you'll hear me refer to them as PSATs. And I have a really annoying animation here, so you're gonna have to hit the button a couple times. So these tags are externally attached. I made this slide like 15 years ago, and I refuse to let it go because I like it so much. So they are externally attached to a fish, and I'll show you the harness that we use for externally attaching them. And when they are externally attached, they collect and archive environmental data. And so for our purposes, they record or they measure and record temperature, depth, and light. And then next, they detach from the fish, they float to the surface of the ocean, and they transmit archive data to overhead satellites. And these are National Weather Service weather satellites. And so the depth, temperature, and ambient light intensity data are transferred to the satellite, and then we can just log on to a server and retrieve those data. And this is really important because, I got ahead of myself. As this tag is transmitting, a highly accurate end location is determined, and it's accurate to about plus or minus 150 meters, which is pretty good in the scheme of the ocean, the Pacific Ocean. And these tags are important for us because they're fisheries independent. So we can put these tags on fish. I'd like to say we forget about them, but we really chew our nails until we hear back from the tag. But we don't have to go recapture the tag. And so if the fish goes up in the middle of the ocean, we still get the information back. Or if it swims over to Russia, we still get the information back. And so we've been using 2 makes of tags. They look like that. Next. Like I said, they measure depth, temperature, and light. And we're not real creative, I guess. We call it a backpack harness system. So this is a dolly Varden right here. You can see a human hand there for scale. The dolly's head is down. The tail is up. It is placed in a custom fabricated cradle that's full of water. So it's pumping water over its gills. We blindfold the fish, which typically reduces fish from, fish don't struggle as much if they have a blindfold on. We secure them with highly technical tiling sponges from Home Depot to keep them secure. And then the backpack system consists of 2 bars that we put at the bottom of the dorsal fin, and we run it's essentially a horseshoe-shaped wire through 2 holes in the first bar, through the musculature and the fin ray supports in the fish, and then out through the 2 holes in the other bar, and then twist it on the opposite side so form a secure attachment to the fish. And then we put it through those fin ray supports so the hydrodynamic drag of the tag doesn't pull the wire through the muscle. We've been tweaking this system for 13 years now. It seems to work pretty well. Once the tag releases, the harness still stays in the fish, so we could actually get a 3rd piece of location data. And we put our names on one of the bars so people can call us if they recover one of these fish that doesn't have the tag. Next. And so we started doing satellite tagging research on salmonids in 2012. And our latest deployment was in March of 2025 in Craig, Alaska. We've done over 25 field campaigns. We've done over 600 days of field work. And each of these triangles is our tagging locations, color coded, although I don't see color coded. And they are. Yeah. Okay. Sorry. So up north are Dolly Varden, Steelhead are on Prince of Wales, Yakutat, and Kodiak, and then the rest of the triangles are. So I'm going to walk you through our tagging locations and hopefully put up some pictures to inspire everybody here. But these are for each of the 3 species. And so we've tagged 116 Dolly Varden from 2012 to 2017. We started over in Northwest Alaska in the Willet River. We expanded to Kinnelina Lagoon. And then a couple of years after that, we expanded over to Northeast Alaska and tagged fish in the Congacut River and out in the ocean. outside of Kaktovik. And then we got invited to collaborate on one of the coolest projects I've ever done over in the Rat River and Big Fish River over in Yukon and Northwest Territories, which was quite an honor to get invited over there. That's the biggest dolly martin we ever tagged. It's 95 centimeters long. That was in the Willick River. Some of you may recognize that person. That's the Northwest and North Slope Area Management and Research Biologist for Fish and Game, holding that fish. And then this is a picture of Michael, my co-conspirator in this work, releasing the dolly martin. No, Kibalina, sorry. Okay, next one. So Chinook salmon, we've tagged the most of the three species. We've tagged the most Chinook salmon, mainly because the U.S. Navy is really interested in what king salmon do out in the ocean, and so they've been a really good partner to work on this with. Each of these triangles is where we tagged fish. We started in 2013, and we continue that work to this day. We've tagged almost 225 of them, and I think our favorite field location, this is Castle Cape outside of Chignik, Alaska, over in the lower left. And we had some great fishing there. And then this is the biggest king salmon that we've tagged so far. And we did a length girth calculation. We think it's about 45 to 50 pounds. And it was, I don't know, for me, it was interesting because these fish are very few and far between now. So the average size of Chinook salmon declined quite a bit. So it's quite a treat to see this big fish. Next. Okay, and then steelhead. So we started steelhead tagging in 2018, and we continue that to this day. We've tagged fish in the Karluk River, Seattle River, in Yakutat up in the middle there, and then in 3 river systems in Prince of Wales Island. This picture on the lower left is one of the rivers on Prince of Wales Island in which we tagged fish, and then this is a tagged steelhead. So yeah. Okay. So then we get all this data, and we scratch our heads and say, okay, what the heck are we going to do with all this data? And so we are mainly interested in movement for a lot of the stuff we do. And so what we do is first look at end location. So that's where the tag pops up to the surface, reports the satellites. And if we can have 5 tags with the same pop-up date, we can look at sort of small scale or small sample size distribution, which is very informative, but it doesn't tell us about what the fish did between release and courting. And so we use a fancy statistical model to estimate the most likely movement track of individual fish. And so this involves matching. I'll give you 3 lines of information here. So if a fish dives to 300 meters, that means it has to be so far offshore because it's too shallow to dive to 300 meters inshore. So we can exclude all of those locations as potential locations for a day. Then when a fish goes to 0 meters and reads a temperature reading at 0 meters, and we'll say it's 15 degrees Celsius, we can look at a sea surface temperature map for that exact same day and say, okay, these are the only places where it was 15 degrees Celsius on the day that the fish measured 15 degrees Celsius. And then we can use ambient light intensity. And what we do is get time series for that, so it's like low light intensity during dark. You get a sunrise event where the light intensity increases. You get a sunset event where it decreases. And you can identify sunrise and sunset, divide that by 2 and get local noon. And that is and compare that to Greenwich Mean Time, and you get a longitude estimate that way, much like people did before they had GPS. And then certain times of the year, you can estimate latitude by day length as well. So you can't do that around the equinoxes, but you can do that around non-equinox period. So we put all 3 lines of evidence information into this fancy statistical model, and it gives us points like you see in this panel up here. And these are estimated daily locations. They're good for plus or minus 25 kilometers. And so they're not good for fine scale work, but they are great for large scale work. And then these are color coded by month. And so you can see this was a steelhead that swam from the Seattle River and swam out to the Aleutian Island chain. And you can see that it started in June and its tag popped up, and it's August, I think it is. And then we do this for a group of fish, and then we look at whatever the human activity of interest is. We superimpose tracks on top of different maps of human activity, whether that's oil and gas lease areas or naval activity areas. And then so hydrocarbon leases, marine protect. We've done some MPA work, Navy training activities, and then when they're in a certain area, we're interested in their diving behavior, particularly in the realm of like bycatch mitigation. So if you're trawling at 200 meters and your salmon are at 200 meters, you're going to catch it with bycatch. But so perhaps in the future we can make recommendations on tailoring trawl depths to avoid salmon. And so we look at this information. So this is a time series of data from mid-June to mid-August. Each dot here is a depth reading by this steelhead, okay? And then each dot is color-coded by temperature in degrees Celsius. And so we can get an idea of how deep the animal dives and then what the water temperatures were that it occupied. And then we can aggregate these data among all the individuals, get sort of group level information about things like night versus day depth differences, and seasonal differences. Okay. This is something that we did not set out to do, and now it's one of our main research thrusts. So we can study mortality as well. And so picture a beautiful free swimming steelhead swimming along the Aleutian Island chain, and a salmon shark swims up behind it, lunges, grabs it, and the tag grabs the whole fish and swallows it, tag and all. So then the tag is in the animal's stomach, okay? And the tag was reading ambient water temperature, but now it's reading the stomach temperature of the predator. And it's also measuring the depth of the predator because it's in the predator's stomach. And so what we look for is abrupt changes in the temperature measured by the tag, the depth-based behavior, so diving pattern of the tag, and then light data. So if the light goes from, nice bright sunlight and then it goes dead dark, then that means it's in the predator's stomach. That's our attempt at humor. And then the most likely predator is inferred by stomach temperature, behavior, and known distribution. So we look at what the temperature is, what the diving behavior is, and what the distribution of potential predators are, and make the technical term is inference. It's a guess based on information. We don't know, except for one animal, we have a pretty good idea. But the others, we are making our best educated guess. So getting into some results here. So the pop-up tags have provided over 14,000 days of data for aggregated among Dolly Varden char, steelhead, and king salmon. Much of the data comes from king salmon because that's the fish that we deployed the most tags on. Dolly Varden have fewer days of data because we've tagged a little bit less of them, but we also use very short deployments on Dolly Varden because they don't spend much time in the ocean. And the tags need to be in salt water to communicate with satellites. So we're forced into making the tag pop up when the animals in salt water. And then steelhead is sort of in the middle there. We have 3,000 days of data from steelhead. So this represents 13 years of our hard work right here. And this is individual tracks of every animal that we've ever tagged and got tagging information back from. They are the white circles, the bigger white circles are end locations or where those tags reported to satellites. And then the dots, the individual small dots are estimated daily locations. And then they are color coded by the species. So Chinook salmon, Dolly Varden, and steelhead. And the take-home messages are that steelhead and Chinook salmon swim a long way in the ocean. So we have fish regularly move more than 3,000 kilometers in the span of 2 or 3 months, and we've had one king salmon swim like 5,500 kilometers while the tag was still attached to it. Dolly Varden don't swim as far, but that's also a little bit biased because the tags aren't attached to them as long either, so they don't get a chance to swim as far with the tag attached to them. And then all species are found both in nearshore and offshore areas, but steelhead are definitely the most sort of oriented away from the coast. Steelhead spend most of their time swimming along the continental slope, which is the transition. So the continental shelf is the relatively flat area near the coast. And then at about 200 meters water depth, you get a steep transition, and that's the continental slope. And that goes down to the basin waters that are like 4,000 to 6,000 meters deep. So on the order of 3 to 5 miles deep. And steelhead seem to really like the continental slope for whatever reason. Chinook salmon generally like the continental shelf, and Dolly Varden generally occupy the continental shelf. So we're going to look at end locations and movement tracks by individual species here. And we'll start with Dolly Varden. And I just lied because we're not going to look at individual tracks because our tags are on the dollies so short that we don't have the error of the individual tracks in some cases is greater than the distance the fish moved. And so it's not informative to show movement tracks where the error is actually greater than the end location. And so we're just looking at end locations. You can see over in the Chukchi Sea off of Northwest Alaska, those fish swam offshore, and most of them swam over north of Russia during the summertime. Interestingly, the area that those fish swam to and their tags popped up seemed to be loitering in that area, which is also a very well-known bowhead feeding area. And bowhead whales like to feed on krill, and dollies like to feed on krill when they're in the ocean. And so I think they are essentially in a sort of feeding hotspot where whales, seabirds also occupy that place. So they're in a feeding hotspot where bowhead whales, seabirds, and dollies seem to sort of aggregate in this area for feeding. The Congacut fish swam offshore, and what I don't have on here is ice extent. And those fish seem to chase the ice as it broke up and moved north. And so those fish seem to be oriented with the ice edge. The ice edge is known to be very productive and has a lot of phytoplankton falling out of it that sort of spurs the food chain. And so there's a lot of zooplankton food around that. And the dollies seem to be sort of following that ice edge as it migrates, as the ice melts and the edge migrates north. And then these Mackenzie River area fish were largely oriented around the Mackenzie River plume. And I don't know if anybody's ever seen that, but it's like chocolate milk pouring into clear water if you look at a satellite image of it. And it forms this very distinct edge. And those Mackenzie River fish seem to be really sort of feeding around that edge. And a lot of fish feed around features like edges because that's where there's a lot of food productivity. Next up is Chinook. And these are end locations in white color coded, or most likely daily locations color coded by month. And then I've got some extra bonus polygons in here too, including the Navy Temporary Maritime Activities Area, the Navy Western Maneuver Area, which is this big opaque polygon. And then I'm going to talk about this opaque area here in a little bit. But you can see that Chinook salmon swim all over the place. They're actually really difficult to study because they don't really school. They travel in very loose aggregations. They hang out at feeding areas, but some of those fish swim north, some swim south, some stay for a month, some stay for 2 days. And they are pretty much impossible to make generalizations about because there's so much variability within their behavior within that species. But you can see they are largely a continental shelf animal. They largely like these shallower, closer to shore areas. They generally hang out in water depths that are like 300 meters or less, except when you get, has anybody seen the old Tom Cruise movie, The Minority Report? That was a good movie, right? This is the minority report here. This fish left Dutch Harbor in the middle of the winter, went out to the middle of the ocean, and did not get eaten by another animal. And it did something completely different than all the other fish did. So they have a lot of variability. Okay. And then steelhead. Steelhead have much less variability. So far, every steelhead we've tagged, no matter where it is, swam west of the Aleutian Islands. And so the Prince of Wales fish, they cut across this ocean basin where it's like 3 to 5 miles deep. And then the Seatuck River and Carlick River fish get on that continental slope, which is that, it's anywhere between 300 and 4,000 meters deep. And they seem to hug that slope. They get out to the Aleutian Islands and start sort of doing donuts around the islands feeding. Or I assume it's feeding. And then the minority report for steelhead is this fish that swam up around the Pribilof Islands. And I have no idea what that fish is there. And we reran the model like 10 times because we couldn't convince ourselves that fish actually swam up there, but it did. So steelhead have much less variability in terms of migratory direction and distance than Chinook salmon. So, okay, so depth by species. I was trying to make this as ungeeky as possible. So I was trying to think of like, how do I show this data that's somewhat interesting? So this is aggregated data among all 3 species, okay? And these are called box plots. And the thick line in the middle is the mean depth occupied by each species, okay? The box contains 50% of the observations. And then the lines, these vertical thin lines for each contain 95% of the observations. And then the dots are what are outliers essentially. And then the vertical axis is depth in meters. So, and a meter is about a yard. Or that 2 meters would be a fathom for people who operate in fathoms. And you can see the Chinook salmon vertical axis goes down to 500 meters. The Dolly Varden vertical axis only goes to 100 meters and steelhead to 150 meters. And so you can see that Chinook salmon are a relatively deep diving animal and regularly occupy fairly deep water depths. And Dolly Varden and steelhead are almost primarily surface oriented, barely make any deep dives whatsoever. Like a deep dive for a steelhead is deeper than 50 meters, and a deep dive for a Dolly Varden is 25 meters. And then if we had all night, we could tease this apart on multiples like seasonal scales and day versus night. But we don't have all night. So I had to try and come up with some sort of meaningful comparison. I did for each of the 3 species that have some means and some standard deviation, which is just a measure of the variability. And so Chinook salmon, occupy deeper water than steelhead and Dolly Varden and also have more variability, and they also occupy greater maximum depth. So we've had some Chinook salmon go down to like almost 550 meters. Okay, next one. This is the same type of plot, except for water temperature occupied by the 3 species. So Chinook salmon, Dolly Varden in the middle, and steelhead on the right. These are box plots again. So the thick line is the mean temperature that these fish are occupied. The box contains 50% of the observations, and then the line's 95%. And the take-home message is that steelhead occupy a very narrow band of water temperatures in the ocean. And this has been shown in previous research as well. And Chinook salmon and Dolly Varden occupy a much wider range of water temperatures when you're in the ocean. Yeah. Sorry, Anna. And then mortality. So this top panel here, I'll walk you through. So the top panel of the top panel, if that makes any sense. The black line is occupied water depths by a Chinook salmon. And so that's from 0 to 300 meters deep. From December, it's about a month. 2 months from December to February. And you can see this happy Chinook salmon swimming around the Bering Sea, feeding, diving, doing everything that a Chinook salmon typically does. Okay. And then the water temperature is this blue line down at the bottom. So that's 6 Celsius, which is a little under 50 degrees Fahrenheit. Okay. And so it's like it's constant temperature water, it's well mixed water, so there's not like a thermocline. And then suddenly, can't really see it that great, but the water temperature or the temperature measured by the tag jumps up to 26 degrees Celsius, which is like 71, 72 Fahrenheit. Okay. So we looked at that, and you can also see the tag starts going to deeper depths as well. So I first looked at the temperature and I was like, oh, this has to be in a fishing boat. Like it's up on the bridge. Somebody caught this tagged fish and it's riding around in their fishing boat. But then we looked at the depth and it's measuring 26 Celsius or 70 degrees down at 300 meters water depth. And I was thinking to myself, where is it 26 degrees at 300 meters water depth? And the answer is in a salmon shark's belly. And so here's a salmon shark. Here's a fun fact. Salmon sharks are named salmon sharks because they like eating salmon. And so this is an example of the inference we make on what's eating our science project, like that other paper said. And so we're able to parse out predation events to warm-blooded sharks, which are essentially salmon sharks, cold-blooded sharks. So we get a lot of steelhead that are eaten by blue sharks, which are increasing in abundance in Alaska. And I don't know if blue sharks are actually increasing in abundance range-wide or they are moving farther north into Alaskan waters, but they were very rarely encountered like 20 or 30 years ago, and now they're frequently encountered southeast all the way over to like Yakutat. And the Yakutat, we charter boats to do all of our field work, so I'm always quizzing people about what they encounter. And in Yakutat, they regularly encounter blue sharks now on almost a weekly basis. And then we think we're able to infer pinniped predation, so that's stellar sea lions. And what we do is get a tag temperature that's 36 Celsius, which is like 98 degrees. And then this is kind of funny, a stellar sea lion eats a big king salmon, and then the depth reading is 0 for like 3 days. And we think this big fat hapless stellar sea lion's eating a nice meal and then goes and just sits up on a rock and digests. We think we're able to infer toothed whales, and that's killer whales, because their stomach temperature is the same as a stellar sea lion, but they continue to dive and forage afterwards. And they have a characteristic diving pattern that matches what we see in our tag readings. And then we have some unknown predators. They are a cold-blooded fish that's associated with the bottom. And the only thing we can think of is a really whopper halibut or a sleeper shark. And unfortunately, sleeper sharks and halibut have very similar diving behavior, and so we can't parse those out. But the predator assignments vary by region. Salmon shark predation is really common from Kodiak west. It's really common. There are apparently a lot of salmon sharks there. We do get some further east, and then marine mammal predation becomes a little bit more common east of Kodiak, but not to nearly the level of salmon sharks. Salmon sharks have a sweet spot for Chinook salmon. They eat a lot of our tagged king salmon. And then this unknown mortality is most common for steelhead and dolly varden. And those are like bottom fish of cold-blooded bottom fish of some sort. So we're getting towards the end here. So pop-up satellite tags offer an effective platform for studying several aspects of ecology of large anadromous salmonids. They've sort of, I don't want to say revolutionized, but they've really advanced our knowledge of the ocean ecology of these species and provided a lot more fine-scale detail than previous research. And they really, one's not better than the other. They each provide different sort of lines of evidence, and so they're really nice complementary tools to use. Next. So for dolly varden, this was the first documentation of dolly varden occupying offshore waters in the Arctic Ocean. And so So in fisheries, we have a lot of biases. So if we only sample nearshore waters, then we make a conclusion that Dolly Varden only occupy nearshore waters, and that's because we weren't sampling offshore waters. And so these tags have been really good for showing us that they actually do swim quite far offshore. We've had some of them swim 400 kilometers from land. These offshore areas are likely important feeding areas, especially that feeding hotspot over north of Russia. And there was in the Chukchi Sea, so in northwest Alaska, there was no evidence of Dolly Varden occupying hydrocarbon lease areas. And so these little tiny dark squares in that top panel are the lease areas that were really big headline news in like 2000 to 2012. And suddenly there's been a lack of interest on that topic, but we didn't get funding to study these Dolly Varden specifically to find out whether they occupied those lease areas. We didn't have any evidence that they did. But over in Canada, so here's Alaska, here's the US-Canada line. If anybody's seen this, it's pretty cool. It's a big clear-cut swath that runs right through the middle of the wilderness. It's amazing. Parker's hiked up and down it. But these shaded polygons are Canadian lease areas, and Dolly Varden do occupy lease areas over there. And there's a big MPA over here as well, and Dolly Varden by that marine protected area as well. And we do get certain benefits from that MPA while Dolly Varden are occupying it. And that MPA largely prevents industrial shipping traffic should the Northwest Passage get established from passing through that area. So, So steelhead, steelhead originating in Alaska generally follow the conceptual migratory pathway to common foraging areas around the Aleutian Islands. That's not new. And that had been found in previous research, but people did not know if the kelps, so the post-spawn adults, whether they actually swam that far offshore to occupy areas as the virgin steelhead did. And they do. So they swim far west, which is more a further migration than previously assumed. And what we've done on this figure is overlay oil and gas lease areas in the Gulf of Alaska and Aleutian Islands as well. And boom, so Bureau of Ocean Energy Management funded this as they were interested in some oil and gas lease areas in Cook Inlet and adjacent to Cook Inlet. We're curious about what fish would occupy those. And steelhead distribution seems to be influenced by this fairly narrow thermal window. We don't have any observations of steelhead outside of 5 to 15 Celsius, which is like 50 to 65, a little under 50 to 65. And 90% of the time, they're even in a narrower range than that. And we think this temperature, this thermal window may also relate to their shallow vertical distribution. So they spend almost all their time in the top 15 meters of the water column, which is the warmest part of the water column. And so we think that their distribution is maybe limited by thermal constraints. Next. And then king salmon. King salmon are a huge challenge to study. And we keep putting more and more tags on them, and then it makes it harder and harder to draw any conclusions because they keep doing new things, which is great for me because I'm really curious about this. But eventually, the funding agency is going to stop giving us money because they're going to be like, okay, you've done enough Chinook salmon research. But they have highly variable movement, and we think some of this is because there's different fish of different stock origins do different things in the ocean. And then we're also studying some fish that are immature and won't spawn in the next year. And then in contrast, we're studying some fish that are maturing and on their way back to their natal river. And so it's sort of an apples and oranges comparison, comparing the migration of a feeder king to a maturing king that's returning to its natal stream. Every single Chinook salmon that we've tagged, all 223 of them, have originated in Oregon, Washington, British Columbia, or southern Southeast Alaska. And so we have never tagged a fish with natal origins north of southern Southeast Alaska, and that's because it's a numbers game. There's so many more Chinook salmon being produced in the southern portion of the range, especially with hatcheries down in Vancouver and Washington, Oregon, compared to only wild fish production in Alaska that is just dwarfed by the hatchery production. Yeah, no, that's okay. Yeah, we take fin clip samples and then have genetic origins. And I should have mentioned that. I'm glad you mentioned that. And then occupancy of offshore waters may be more common than assumed. We do get fish out in these offshore areas. And then buried under here, I told you I'd revisit it, is that U.S. Navy temporary maritime activities area. And can you go forward? So I've removed the fish track so you can actually see it. This polygon is the TMAA, and fish were swimming through there. And so the Navy said, well, when they swim through there, where are they? And we said they're almost primarily up on the continental shelf and slope in this opaque area. And so, and this was shocking as a fisheries researcher because our recommendations don't frequently get incorporated into management decisions. And the U.S. Navy closed down explosive activities over that area to reduce their impact on Chinook salmon. So, yeah, can you believe it? Okay, next. So, Chinook salmon and steelhead mortality is common, widespread. It occurs year-round. And it seems to be mainly pinheads and salmon sharks with a little bit fewer killer whale predation. We do have some killer whale predation. And then this is a big question in my mind. I am not making any bold proclamations here, but both of these species are struggling. And we've learned that much of this struggle is because of dangerous conditions in the ocean. And so I sort of wonder whether the mortality that we see, these fish getting eaten, is merely a coincidence. Is it related to this high level of predation, or the decreased abundance? Is it related to the high level of predation, or is that just a coincidence? And I think for me, what it points out is we need to take this more seriously, because traditional fisheries viewpoint is that the first ocean entry and the first winter are the critical bottlenecks in abundance. And people ignore the rest of it and say, oh, you know, year class strength, so how abundant a fish is set at ocean entry and at the end of the first winter, and then after that, these fish are relatively safe. What this points to me is that they might not be so safe after all as older, bigger fish, and I think we need to start doing more investigation. Next. And then so what I've shown you is mainly sort of descriptive, like averages, variability, maps. We are using these for much more sophisticated analyses now. And I won't bore you with those analyses. But what we're doing is we're trying to understand Chinook salmon predation on a regional scale. And so we're actually looking at whether there's size selectivity related to season and predators. And we should have that done fairly soon. And then we're looking at environmental drivers of behavior and distribution. So we're looking at, are there certain sets of environmental conditions that fish are typically associated with? And if there are, could we use that for leveraging like bycatch mitigation strategies? So we're trying to, like the steelhead thermal window is a good example. Like if steelhead occupy a really narrow thermal window, could we use that to our advantage to avoid them in the ocean? And then we are also, and this is really gaining traction quickly, using the information, particularly on depth, to understand susceptibility to bycatch and informing fisheries practices. So we are trying to tease out patterns in depth distribution by season and location, and then work with trawl fleets to compare the depths at which they fish to the depths at which salmon occupy, and see if we can separate those to mitigate bycatch issues. So these are all works in progress right now, and we hope to have a bunch of this work done over the next year. So if I'm invited back, I'd be happy to talk more about it. And then future direction. So this is what we're planning on doing in the near future. And I've broken it down by species. Steelhead's going to come last. So we'll start with Chinook salmon. So, and I divide Chinook salmon by two sizes. They're either big or they're small. And so I'm talking about the big ones. And we have spatial and temporal gaps. So we have released fish in certain locations at certain times of the year. And so our data are not uniformly distributed throughout the year and in space. And we are trying to fill those gaps by tagging in more locations. And then we are starting an acoustic tagging project. So I said the title of this is a little misleading because I said salmon and satellites, and this is not a satellite tag. This is an acoustic tag, which is about the size of your finger. And once a minute, it sends out a ping and ping, ping, ping. And this fish swims around. And this is Michael doing a surgery. It's a little blurry, but those are sutures in the fish. They're abdominally implanted and they stay in the fish for the rest of its life. And there is a series of underwater microphones called hydrophone receivers. And each of these circles is a hydrophone receiver that's underwater. And when a tagged fish that is pinging swims by a receiver, it records its presence. And so we are collaborating with people in Washington, and the Navy has a training range down here off the coast of Washington that they're interested in learning more about these. And so we are doing a field deployment. We're hoping to tag 100 king salmon out of Craig, Alaska in March of 2026. So that's the future work in the near future for big king salmon. Next, this is almost brand new. Now we're talking about small king salmon and UAF. I'm fortunate I was invited to partner with Alaska Department of Fish and Game, and we are implanting miniaturized acoustic tags. So these things are like the size of a pet tag that you put in your dog or pets, you know, when they scan them and so you can tell your pet at the pound if it runs away. We are putting these in king salmon smolts that are about 9 to 10 centimeters long in the Kenai River as they're making their way to the ocean. So that's a Kenai River Chinook salmon smolt with a tag above it, right above its dorsal fin there. That is 10 centimeters long or 4 inches. And that is a Fish and Game employee who's also a graduate student at UAF, and she is implanting, and this is actually a Kenai River Chinook salmon smolt right there, that silvery thing. And she's implanting one of those tags in it. And then as they make their way down the river, hydrophone receivers, this one's high and dry because it's low tide, but if you imagine the tide coming up, this thing would be floating in the water, and a smolt swims out of the river, goes into Cook Inlet and hangs a left or a right, and it will swim by one of these and it will record its presence. And so we can understand how long they loiter in the area, which direction they swim, and how they survive when they hit the ocean. So we did our first field season this past year, and we have 2 more field seasons coming out in 2026 and 2027. We are also expanding into chum salmon. So this is work being done by an Aleutians East Borough employee who's also a master's student in fisheries at the University of Alaska Fairbanks. And she is putting a satellite tag on chum salmon here in the very controversial area M fishery in the Alaska Peninsula, trying to answer a bunch of questions directly related to management, and they're getting good information out of this. Here's Charlotte throwing gently releasing head first a chum salmon over the side of the sailboat on which she tagged that fish. So this is exciting, and we are hoping to do more chum salmon tagging to answer it. They are a very hot topic right now, and there's a lot of issues surrounding chum salmon, and so we're hoping to provide some information about them. Next, my picture didn't show up. Darn it! I had 2 pictures here, but apparently my PowerPoint doesn't like it. We're collaborating with a group in Vancouver, British Columbia, and they are trying to answer the question of how many sockeye salmon that are susceptible to seeing fisheries in the north end of Vancouver Island actually make it to the Fraser River, and how many of them get eaten by different predators. And the The assumed culprit, this is really interesting, is the Steller sea lion. And everybody said Steller sea lions are eating 40% of the salmon run. And so everybody's been pointing their finger at Steller sea lions. Well, it turns out that Steller sea lions don't eat that many of them. And salmon sharks are also highly abundant around it. Eat a lot of next. All right, here's my picture. And then closing on steelhead, we are hoping to expand our tagging research into other river systems. And so I have the Anchor River Weir and then the Nelchik River Weir. We are partnering with Fish and Game this coming summer. We have some satellite tags that we want to attach to steelhead in Kenai Peninsula rivers. We don't have a firm plan right now, but we are hoping to partner with we are partnering with Fish and Game. We're hoping to partner with other organizations and sort of fill in some geographic gaps in our steelhead tagging. And the reason why is, oh man, I lost another slide. I had another slide, but apparently it disappeared. So, we are. I'll give you the punchline. We are collaborating with groups that are doing similar steelhead research in California. 3 rivers in Oregon, 2 rivers in Washington, 2 rivers in British Columbia. And we are trying to get a holistic, range-wide examination of steelhead migration, behavior, and predation throughout the range in North America. And so we are slowly, we are doing this in sort of a piecemeal fashion because getting dedicated steelhead funding is much more challenging than fish captured in commercial fisheries. And so we've tagged in Alaska, in Prince of Wales, Yakutat, and Kodiak, and we are hoping to tag in the Kenai Peninsula, and then in future years move further out in the Alaska Peninsula. So that's what we're envisioning, and we're hoping to make that work. Okay, this has been a ton of work, and we've had funding from a bunch of different funding agencies. Most of the steelhead tagging has been funded by Bureau of Ocean Energy Management. The king salmon tagging, most of it's been provided by the U.S. Navy, some from Pollock Conservation Cooperative, and then some other organizations. And then we have, on almost every project, we've partnered with Alaska Department of Fish and Game. They've been a great project partner, and we're always looking for additional project partners. I think there's just a slide. This is everybody who's helped us. You don't have to read all the names unless you want. And then we have there's one slide. And this is the publications that we've written on all this work. And if anybody's interested in any of these, I obviously don't expect you to read all these right now, but if you're interested in publications, let me know when I can see PDFs of them. And that's it. Okay. It's got a tungsten pin in it. That's like a piece of dental floss. Tungsten is stainless in salt water until you run a current through it and change its electrode potential, and then it corrodes for like 10 minutes once you put some battery in it. So there's a battery in the tag, and it just runs a current through a tungsten pin. The pin corrodes, and then releases.
What determines that?
There's a battery, and then the tag engineering does.
You can just push that like a button.
We program it ahead of time. So the tag has a user interface that we can program a bunch of things into, and we program it. Yeah.
You said that almost all steelhead only spawn once. So most of the steelhead that you're finding in rivers are spawning for the first and only time.
First time spawners, yeah.
And they're not
by number. We do get some second time spawners, those are the really big ones, and very rarely third time spawners.
really. Yeah. So what's life expectancy? So after they spawn for their one and only time, they just go in the ocean about the rest of their life.
They this is a guess.
Yeah.
They go to the ocean and figure out it's really dangerous, and they're weak and delirious and adjusting to salt water, and they get eaten by something. So yeah. Yeah.
Yeah.
Yeah.
So any
Chance of doing pop-up tags on, like in Inlet.
So in 2016 and 17, we tagged 40 Chinook salmon out of home. And we did short-term tags where we were answering a specific question about salmon shark predation there. But we would love to revisit that because we really only have data from March.
Would you expect them to still go west?
Chinook or steelhead? Some of them, yes.
I've heard some of them go like
Yeah.
Canada.
Yeah. Yeah, and so conceptually, Chinook swim out of their natal river no matter where they're from, and they hang a right, and then do a loop. So Gulf of Alaska, they do a loop, and then return home at some point. And there's a lot of variability in the details of that. But conceptually, they all do that sort of loop around. So yeah.
Can you repeat the question for folks online?
Oh,
But going forward.
Okay.
Yeah, thanks.
I got another question.
Yeah.
Dolly.
Yeah.
This is fascinating. So the dollies, you did all the tags were up there.
Yeah.
Have you, and they have the same like history, like dollies in the south, all come in and spend their time in, they overwinter or we don't really know.
So southern, so the question was, what do we know about differences between northern dollies and southern dollies? So southern form dollies are kind of a different beast. And so there is a northern form, which is north of the Alaska Peninsula, and a southern form, which is south of the Alaska Peninsula. And they actually have a different number of chromosomes. Northern form dollies are very frequently anadromous. And when they're anadromous, we think they spend more time offshore. Southern form dollies, and this is just going on what we've sort of read. People don't know a whole lot, but they think they spend a lot more time close to their river mouth. And so they do go out the ocean, but they don't make any big migration offshore. And they don't get as big, which is kind of crazy because the water's warmer and there's a lot of food in southeast and south central. And the Arctic is a cold place, but they get much, much bigger in the Arctic.
Super fascinating. On residency.
Yeah.
And each place genetically different.
Yeah.
Each river. Hey,
Okay. Yeah. I think there's a question over here. Yeah.
What percentage of your samples lost to mortality?
What percentage of the samples are lost to mortality? Dolly Varden is very few. Steelhead is probably 20%, 25%, and king salmon is like 40 to 50%. Yeah. What's that?
Is that handling mortality?
So that's a question that we frequently ask ourselves. We have no evidence. Most of that mortality occurs like 2 weeks to 6 months later. We had one fish swim 5,800 kilometers in 260 days, and then it got eaten by a salmon shark. And so it does not appear what fish biologists typically assign to like tagging and handling mortality is like the first 2 to 7 days, and we don't have much of that. Now it was track. Yeah.
What kind of data do you have on your tag fish being captured in commercial fisheries?
Very lit. So how many of our tag fish are recaptured in commercial fisheries? Very few of them. We have had, I think, 3 Chinook salmon captured in a commercial fishery at the mouth of the Columbia River in a gillnet fishery, which is kind of interesting. We actually get more tags that release from the fish and wash up on a beach, and a beachcomber finds them. And so we've had probably 10 tags found by beachcombers in like we had one tag found on like Unga Island in the Aleutian Islands. And so we get tags discovered by beachcombers, and it's pretty amazing.
So the predation greatly outnumbers catch by commercial fisheries.
By far. Yeah. Yeah. And that's something we're examining in that paper, is how commercial fishing intensity compares to fishing. Yeah.
About the application,
It doesn't. Does the does the migration relate to the Coriolis force? Yeah, the Coriolis force turns to the right, but the current goes to the right. And then the current also leads to more productive feeding areas. And so I don't know if the migrations because the Coriolis force is making them go that way or whether they're just genetically pre-programmed to go north to find food. Yeah.
Do you have a theory behind the Chinook spending less time in the salt and like seeing them come back to spawn at younger ages now, like more Japs in the system?
I do have a theory. Do I share it? Yeah. Do I have a theory about why Chinook salmon are returning at on average at younger ages to spawn? And they're also, so there's two issues. One, they're smaller size and age, and they also have a smaller or younger age of maturity. Here's my theory, and I don't know if it's right or not, but there's a lot of data to back, some data to back it up, is the ocean has turned into a dangerous place. So here's my two pieces of evidence. One, the Marine Mammal Protection Act put the kibosh on killing marine mammals, and there's a lot of orcas and sea lions in the ocean compared to what there was in the 50s, 60s, and 70s when we actually started counting fish in earnest. So I think we started at probably a high point. I think there's no evidence to support this other than inference. I think there's way more salmon sharks in the ocean than there used to be. And that claim is based on, and I don't say this very often, but I'm an R, so I can say what I want. There used to be high seas drip net squid fisheries, you know, the big like floating curtains of death that were like 50 miles long and they caught everything in the ocean. They used to catch 30 to 50,000 salmon sharks a year for dozens of years with no signs of decline in catch per unit effort. And so salmon sharks were somehow resilient to fishing pressure in that. And now that fishing pressure has been removed, I think there's probably a lot more salmon sharks in the ocean as well. And so anyway, fish that spend, going back to the question, fish that spend longer in the ocean get eventually. And so what you get is selection for fish that return to spawn at younger ages because they are less susceptible to danger in the ocean by the mere fact that they're out there for a shorter period of time. So that's my theory. Yeah.
Is anybody doing salmon shark population studies?
No. We are doing some salmon shark tagging. So I partner with a person in the Alaska Department of Fish and Game, and we're doing salmon shark tagging. Counting salmon sharks is really challenging. And so without some giant dedicated effort, it would be impossible to get an abundance estimate of salmon sharks. But it's I think one of the key questions to this riddle about how many salmon sharks there are. Yeah.
I don't know, have you done this or will you do this to look at with those 2 billion pinks being turned out by the hatchery? You know, I grew up in the country where we had big ranches, and on years where it's dry and you ran out of food, your cattle, you know, nothing survived. They were just so skinny.
Yeah.
So there's only so much food to go around, and we see less kings back, we see less steelhead, and the ones that come back usually aren't very chunky. They're pretty long and lean.
Yep.
So there's got to be. It seems like nobody wants to face the fact of all those pinks out there. Just they're eating machines.
Yeah. There's a lot of convincing evidence that we are at the ocean carrying capacity, and it goes beyond fish. When pinks, you know, the odd even year of pink salmon, the odds are the most common. Seabird hatching success goes way downhill during odd years. Marine mammal twinning rates go way downhill. It reverberates throughout the ecosystem. And so there's a lot of convincing evidence that there are indeed density dependent effects happening in the ocean as a result of more fish in the ocean, more salmon.
One of the problems we have is that commercial, when you go to the meeting, Palm Fish Department of Alaska, they always defend releasing these. And they said the causation is not this and that, you know, that term. And you look at everything University of British Columbia has done, all these other places, and they all circle around and said there is a definite effect of all these pinks in there.
Yeah.
But we can't get Juneau to accept that because
You should get my
They're
coworkers.
in with the fishery.
You should get my coworker to speak about this. He's very passionate.
He understands too, that-
He does. But yeah, there and you know, I sort of made this tongue-in-cheek comment about making management recommendations and people don't listen to it. Fisheries is a really complicated field because there's biology involved, but there's also emotions and psychology
And money.
and money. Yeah. And so it's this like it's this sort of rubric, decision-making rubric that isn't always based on the best science available. And so, like, you know, I've spent a lot of time studying fish, and you can come up with the best study results, and if it doesn't jive with economic forces, then people like flush it down the toilet and they're like, nice try. And so that's what that tongue-in-cheek comment was about. But yeah, and that's what I don't know. Fisheries is a fun field to be in because it's not. It can be frustrating too. Don't get me wrong, but it's a very complex field because humans are involved.
Okay.
But so I teach a class, a 200 level fisheries class, and I talk about ocean carrying capacity. And the example is if you put too many cows in the pasture, what happens to them? And every student, even an 18-year-old right out of high school, says it gets skinny and eventually dies. And I'm like, yep, there you go. That's what happens. So, yeah.
So you talked about with the bigger tank, the satellite tanks, like being able to determine potential predation with your smaller tags on those smaller fish. Is there any way to determine what percentage of those fish are getting?
So not with the tags that we're using, because we're using the less expensive version. There is one of those acoustic tags actually has a pH sensor on it, and it codes the pH. So when it sends out an acoustic ping, it sends out this pinging code about what the pH is that it's measuring. And so the pH in a marine mammal and shark stomach is very acidic and very low, and so which is different than being in the abdominal cavity of a fish. And so there's actually a generation of these tags that can tell you whether they're in a predator stomach or in the abdominal cavity. But we are not using those right now.
Yeah, I've got a question.
Oh yeah, thank you, Donna.
My question in a way reflects to you. I do work with you.
Yeah.
I have two questions.
Yeah.
My first question is you talked about how 3 tags have been caught by commercial fishermen. Do you have like a bias account for that? Because if I was a commercial fisherman and I saw that and bycatch, I'd probably be like, not that I would do that, but like maybe I would throw it back. Like you.
Now I'm understanding your motive. No. So, I'll tell you an example of how we knew one of these tags got captured in the commercial fishery, is that it started reporting to satellites from a house on shore. And this is funny. Our Navy contact was like, just give me the guy's address. I'm going to go get it. And I was like, no way. I had this vision of her sending in like SEAL Team 6 transmitting tag. And I was like, no way. We don't need the information that bad. So we've had fish get captured by commercial fishermen who do not report the tags to us. But then the tags end up reporting to satellites, and we can clearly see that it was. We've had tags report from the bridge of a boat, tags that I put on halibut, and like move around the Bering Sea, or they're in somebody's house, so we would know.
Okay.
So,
and then the other question is we kind of talked about how the captain talked about reporting tags, especially for steelhead. And I'm wondering if you could just kind of give a rough estimate of like how much these tags cost, like if it like the acoustic tags or like just any tags in general.
Okay. Here's the good news. Acoustic tags are like $350 each. Okay, that's the good news. The downside of acoustic tags is that they have to swim by a receiver for you to detect them. So if the fish swims out offshore and there's no receivers out there, you never hear from it again. So there goes your $400 and it's gone. Satellite tags are $4,000. But we almost always hear back. So we hear back from like 95% of the tags. And then in addition, the satellite tags tell you what the fish was doing tells you a daily location, and then what depth and temperature it's occupying every minute. And so if you amortize it out by cost per data point, the satellite tags win by far. So they're expensive. Yeah. Yeah. Now here's the interesting thing about satellite tags. The first satellite tags were made in 1998, and they cost $4,000 in 1998, and they still cost $4,000. So inflation does not apply to everything. Yeah. My favorite. All right, I'll tell you my favorite. It was a tag steelhead. And we don't know if this happened or not. It was inside a cold-blooded predator, okay, for about 2 months. And we think, and it was, swimming up and down, but the light sensor was totally dark. So we think that tag was in the cold-blooded predator's stomach. Then the light sensor started sensing light, but it was still down, not at 0 meters. Okay. And it was swimming around, but it was reading light. And the temperature signature changed because there was no. So when animals change depth, frequently the water temperature changes. Okay. And if the tag is in an animal's stomach, there's a lag. So the temperature change doesn't correspond directly to depth changes. So anyway, the tag started reading light. It was still reading depth, and it immediately started, the temperature changes caught up with the depth changes. And the only thing that we can think that happened is a shark ate the steelhead. The tag made it around the spiral intestine. So sharks have the spiral valve intestine, and they digest really slowly, and it made it around. The shark tried to poop out the tag. The harness got caught in the shark rectum, and the tag was still attached to the shark, but through its rectum. And that is literally the only story that we can think. It's the only story that makes any sense, as far-fetched as it is.
Now I see the light.
Yeah. So, yeah.
How long it takes to digest these tags. So do any of your anomalies, are they because they were inside a predator and the predator moved in a weird non-salmon direction? Didn't you know that?
So almost all of the tags that were eaten only stay in the predator stomach for like a day to 7 days. Most animals are really good at throwing up. Like, have you ever seen Jaws? It's got like a shoe and a license plate in its stomach. Most animals are really good at, for lack of a better term, barfing up foreign objects. And so what we think happens is the predator eats the tagged fish, gets the tag in its stomach, and then gives it a tummy ache, the shark version of a tummy ache, whatever that is, and the shark barfs it up. And same with marine mammals. And so usually the tag is not the predator's stomach very long. And so we don't get any like crazy anomalies from the short time that the tag is in the predator's stomach.
My second question is, I know you don't have tags on sockeye, but maybe you have background information. Are the salmon sharks also eating a lot of sockeye, do you think, or are they avoiding?
I think salmon sharks eat whatever's in front of them. Yeah, in Russia, their common name is herring shark because they eat a lot of herring. And so I think salmon sharks are true opportunists, where they will eat pinks and river mouths. They will eat sockeyes around Vancouver Island. They will eat a tagged Chinook salmon when it swims in front of them. And I think one of the questions that rolls around in my mind is are our mortality rates actually representative of what's happening in the real world of an untagged fish? And I am guessing our mortality rates are a little bit inflated because the tag, there's been some flume tests where you stick a fish in a flume, and they crank up the water speed and see how long the fish can swim and when it gets tired. The tags don't really have any effect on top swimming speed or swimming endurance, but what I think it does is it gives the predator a cue to focus in on it. So like, I liken it to being the unstriped zebra. The unstriped zebra is the first zebra to get eaten in a herd because it gives the predator something to focus on, and that's what I wonder if that's what's happening with their tags. Yeah.
Is there any effort to recover tags after they come back up, and the ones that do get like found beach scum and whatnot, can they be reused?
So we have this, it's a fancy radio direction finder, and I don't know how it got this name. It's called a goniometer. And we have one of those. And so if we had a tag wash up on the beach in Kodiak, and we used a goniometer and went and got it. We can reuse the tag if we get a hold of the tag before its scheduled transmission date, because transmitting requires a lot of power and it drains the battery really quickly. If we get the tag after the tag has transmitted its data, it's dead. But for what, I don't know why they do this, but the manufacturer will give us a brand new price at a brand new tag at half price if we show them that we recovered a tag. Yeah, and I don't know what's in it for them. Like they do it, so I'm not going to do it. Yeah,
I don't know if you covered this already, but how are you catching these fish?
We go on fishing trips. So steelhead and dolly vart, we just go fishing, like in a river. And then Chinook salmon, we charter, usually sport boat charter operators. We pick an area we want to go tag, and we look for a charter boat operator to take us out. We charter.
Fun.
Yeah.
It's rod and reel.
All rod and reel. Yeah. I tried various iterations of other things, and nothing is as good as rod and reel. Right. Yeah.
What's the bycatch still outside of people catching them over the ocean?
Yes, but I don't know how much. And so it used to be mandatory to report steelhead bycatch, and it's not anymore. So there's no good estimates of steelhead bycatch. One time I was doing charter fishing for king salmon in an undisclosed location, and an undisclosed boat came by and said, "What are these fish?" and held up two great big old steelhead. So yeah, they're still being caught.
Well, yeah, I just mean like I know they catch me on that all the time.
That was in a scene. Yeah. I think they get caught in everything except for. Oh. Yeah.
So how do we recover steelhead and kings?
There's no easy answer to that. The evidence points to the ocean being a dangerous place, and the ocean being a dangerous place is caused by a lot of things, changing food webs, changing temperatures, changing predator fields, changing prey fields, and changing all of that simultaneously to benefit salmon is a tricky task. The freshwater portion is really important because if you can't put a lot of fresh fish from freshwater out in the ocean, then they don't stand a chance. And, Alaska is not perfect, but we have relatively intact fresh water. And there's a lot of good work that happens maintaining that intact habitat and restoring, restoring altered habitat. And so I think that is freshwater work is the most tangible action because saltwater work is so complicated. But bycatch, I think, is an important thing. Bycatch is not the smoking gun. Salmon are facing a death by 1,000 cuts in the ocean. And so there is not a single smoking gun that is responsible for the decline. But if everybody tries to reduce their impact, I think it will benefit safety. Yeah, Donnie.
Mind looking back at the channel
Yeah. Didn't know I'm on board.
That straggler. The straggler is fascinating.
Yes.
The minor.
Guy. Don't worry about that one guy. Yeah, I want to know about what's your read on that straggler that went offshore. 'Cause it reminds me, and I'm sure you've been involved with Kodiak black rockfish research and that black rockfish that basically made a beeline to Hawaii. Like, do you think that's a modeling issue?
No.
These are
That fish, it was plain as day. It was feeding up on the continental shelf and it was very irregular. And then it went out offshore. It traveled for like a week. And then I think it hit a prey field offshore that was down at about 450 to 500 meters because it had a perfect diel diving behavior where during the day it was down at 450 to 500 meters deep and at night it was up at 50 meters deep. And that was in the wintertime. And Chinook salmon are known to be big consumers of squid in the deep sea scattering layer. And I think that thing ran into a squid layer out there. I think it was on the way back to the Pacific Northwest to spawn. It was a big fish. It was like a 25-pound fish. It was on the way back, ran into a squid field out there, and then just sort of slowly worked it in that squid field. And eventually it would have ditched it to go back to spawn.
And it just happened to be that the tag was programmed to go off.
Yeah.
Deploy.
Yeah, that was like a March pop-up.
Okay.
Yeah.
Then are you familiar with the black rockfish tag then?
No.
Kodiak?
No, but tell me. And everybody should watch The Minority Report. Because the take-home message is don't ignore The Minority Report. And so
we're struggling with the black rockfish, right? Like we know they're a long-lived species. Males we're finding out to 50, 60 years old, but females we're losing them about age 20, 22, right? Because there's this sexual difference in longevity.
Yeah.
We don't know whether to hide them or kill them when we're doing a stock assessment. Right? Are they alive and they're just hiding, or are they dying off? But there's this road. I don't even know the sex on the species that was tagged, or on the fish that was tagged. But I think it's a female, and it takes a feline, like the straightest line I've seen an animal take tag wise. Just straight south, 200 nautical miles plus offshore.
it was very similar. It's like a snowbird done. It's going to Hawaii.
Every other black rockfish just stays local. There's no
I don't know. Maybe that's right above a seamount. So maybe it's going up there to feed.
Yeah, you should. I don't know if you talked to Carrie Ward, but you should talk to her about that specific.
I will. Yeah. Yeah. Well, thank you, everybody. This has been great. Great.
Hey, everybody, thanks for coming out tonight. Sounds like you enjoyed the show. I'm gonna try and keep bringing more interesting speakers in throughout the winter, and as usual, about May we'll finish up our series for this year. Take the summer off and start again next fall. Whenever you get the emails, if you guys have any comments or something, just at the bottom of the email, just go ahead and hit a reply. It'll come back to me. Don't hit reply all, or you're gonna talk to everybody that's interested. But hit reply. So if you have any questions, comments, things that you are interested in or want to hear about, just send an email back to me and I'll see it and try and bring something like that out or get that information back to you, whatever you're interested in. Other than that, thanks for coming out this evening. Great to see you. See you next month.